Hello, everyone. Welcome to the special lecture series as part of our summer workshop on big data training for cancer research. I'm Min Zhang from Department of Statistics and the Associate Director of Data Science at Purdue University Center for Cancer Research. The lecture series is sponsored by the National Cancer Institute. We also got quite a bit of support from Purdue University Center for Cancer Research. So the summer workshop was originally scheduled on campus and moved online at the last minute. But I think this is working out really well as we were able to recruit a lot more participants and hit a much larger audience for our special lectures. Today, we have three panelists. John helped the meeting setup and all the registrations. He will record the lecture and share the recordings with all of you after the lecture. Now, I would like to introduce our webinar moderator, Dr. Nadia Lamman. Dr. Lamman is a research assistant professor in the Department of Comparative Pathobiology at Purdue. She also manages the Purdue site of the Collaborative Core for Cancer Bioinformatics, which is a joint core for cancer researchers at Purdue and Indiana University. She has a background in biochemistry, molecular biology, and computational biology. Her research interests include single-cell data analysis and data integration. With that, I will hand it over to Nadia.
Thank you, Min. So welcome. We're glad you're here. Just a few housekeeping notes. We're going to leave your microphones muted for the duration of the seminar. And so if you have any questions, just go ahead and use the Q&A function at the bottom of your Zoom screen. If you have any additional comments or questions, feel free to send a message to me or the other panelists. And then possibly in the middle of the seminar, depending on how many questions we get, and then at the end of the seminar, we'll be going over the questions that you submit using the Q&A function. And with that, I'll hand it back over to Min.
Thanks, Nadia. It's my great pleasure to introduce our speaker for today. Dr. Bren Kibbe is the Chief Data Officer for the Duke Cancer Institute and the Vice Chair of the Department of Biostatistics and Bioinformatics in the Duke School of Medicine. Before joining Duke, he served as an acting Deputy Director of the NCI and was the Director of the NCI's Center for Biomedical Informatics and Information. Dr. Kibbe is a recognized national leader in cancer informatics and data sharing. He has been instrumental in efforts to speed scientific discovery and facilitate translational research by using IT, informatics, and data science to address complex research challenges. He has been a proponent for open science and open data in biomedical research and helped define the data sharing policy for the NCI Cancer Moonshot program. Dr. Kibbe is also co-founder of the Cancer Informatics for Cancer Center Society and has been organizing twice-yearly meetings of cancer informatics faculty and leaders from the majority of the NCI designated cancer centers. Well, we have been talking about different types of omics data in cancer research during the past 2 weeks. Today, Dr. Kibbe will present an exciting project, the Human Tumor Atlas Network, which is part of the NCI Cancer Moonshot Initiative. He will talk about how he builds a framework for integrating multi-omics data to understand cancer biology and to improve cancer care. Please join me in welcoming Dr. Kibbe.
Thank you very much, Min, and thank you, Nadia, for the venue to get a chance to talk with all of you. I may have some bandwidth issues, so I may turn off my video if I get a message that you can't hear my microphone properly, but I'll leave it on until I get that message. First, let me turn on my slides and make sure you can see them. And also let me say that although I will talk a bit about the Human Tumor Atlas project, I'll talk a little bit more and give you some more framing about why the Human Tumor Atlas is so important, and frankly, why the computational biology that we're trying to do is also so important. So I'll give a bit more framing, perhaps, than Dr. Zhang mentioned I would give. So first, I want to thank Ethan Cerami at HTAN for some of the slides, and Fred Streitz at the Lawrence Livermore National Lab for a few of the slides as well that I'm going to present to you. I think it's useful when I give a presentation like this to just to explain a little bit about who I am and how I got here. So my degree is actually in chemistry. I got that at Caltech a number of years ago. I did a postdoc in molecular genetics at the Max Planck Institute, working on on RAS and understanding the role of RAS in yeast, the Saccharomyces cerevisiae. And for the last 20 or so years, I've really been much more on the data side of things, thinking about how we manage and analyze data we're generating, particularly in cancer research. The only other thing that's noteworthy is that I did lose 3 of my grandparents to cancer, and this past year I lost my father to cancer as well. Cancer is a very personal thing to me. So I'm going to talk about 3 topics. One that obviously you know I'm going to talk about, and that's the Human Tumor Atlas. I'm going to talk a little bit about survivorship and precision oncology as well. And I see I just got a note that my internet is unstable. I'm going to turn off my video, and hopefully that will help a bit. I apologize for that. So I want to go through a little bit about survivorship, because I think that actually motivates a lot of what we're doing right now in trying to understand single cell sequencing. And it may seem like a stretch, but in fact, it's really all part of the same body of work. So just to remind you, or to tell you if you did this, as of January this year, there were more than 17 million cancer survivors in the US alone, and that it's estimated that by 2030, there'll be more than 22 million. So that means roughly every year there are more than 500,000 additional cancer survivors in the United States. And that's important because there's a lot of comorbidities that cancer survivors experience. So there's everything from financial toxicity, so the issues that you have fiscally when you go through cancer treatment. It's everything from loss of jobs to just the cost of the treatment itself. Neuropathy, lymphedema, cardiotoxicity, sexual dysfunction, chronic fatigue, cognitive impairment. Of course, you can read all these. The list actually goes on, and that's something we really want to prevent as much as possible. So that brings us to cancer prevention. So if you will, at the very beginning of the cancer journey, how do we stop people from getting cancer in the first place? To doing a better job of providing more effective and less toxic therapies, and of course, more cost-effective therapies. I just saw that Dr. Ned Sharpless, the director of the NCI, tweeted this morning that one of the things he's really concerned about today is the delays that many cancer patients are going through in getting their treatments because of COVID, and that this may have additional complications and lead to poorer outcomes for cancer patients who are going through therapy right now. So it's also important to think about cancer from the complex biology that it represents. So if you will, the promise of precision medicine is really understanding how we can untangle the complex mixture of genetics, epigenetics, the impact of nutrition, the impact of environment, and how those aspects of who we are relates to the clinical presentation, and how that changes the way that we should provide effective therapy. So, and this obviously has many ramifications throughout healthcare. Of course, we're really focused here on the computational biology and how we think about omics data in particular. But I think it's really important to step back and realize there is this much bigger picture of healthcare and cancer in this country and in the world that we also want to take into account. So for today, at least, I'd like you to have as your take-homes that the Human Tumor Atlas Network is really a deep scientific dive into understanding cell diversity, so what cells are present in a given tumor, the interplay between different biological systems. We'll talk a bit about the immune system in particular, and how that may play a role in regulating and potentially even promoting parts of cancer. Population diversity. So how do we make sure that when we come up with a therapy that we understand the differences in effectiveness and efficacy across different populations in our country, in our world. And then of course, understand disease progression. And something, this is more of an editorial, I would say, is that single cell techniques, whether we're talking about things like MIBI that are looking primarily at protein, or we're looking at RNA-seq, we're looking at really detailed imaging modalities that let us look deep into a single cell. These are really changing our view of biology. And really importantly, it's not just snapshots, it's actually starting to understand the dynamics of biology. And to make that possible and for us to actually do those things, we need data science. And it's everywhere all around us. And of course, it's in our science. And something I want to go back to, and I'll hit on this topic a number of times, is understanding the patient context, the patient trajectory as they go from really throughout their entire life. So all of us going through our lives, understanding that trajectory is important. And of course, for cancer, it's incredibly important. So again, since all of you are here for a big data course, I don't think I need to talk very much about this. But technology today has radically changed. You now have devices everywhere. Smartphones are ubiquitous. We now have terms like smart homes, smart cars, smart cities, and they're beginning to mean things. So you can now, of course, and this has been true for a number of years, you can load Google Maps and you can see really real-time traffic patterns almost anywhere in the world. Real-world evidence, though, is basically doing that same kind of thing, but let's do that with our health records. So how do we see what a given patient has gone through for their treatment, for their outcomes, their trajectory, and how do we use that to really inform the next generation of healthcare? Likewise, built environment data. So that's all the data are all of the environments that are around us. So it's toxicity, it's the presence of good food in our environments, the ability to walk. So it's also mixed in with social determinants of health. Precision medicine and learning health, again, that's another way of taking what we're doing and really applying it to building these learning health systems for cancer. And then again, coming back to single cell measurements and data science. So all these things are really combining in, I would say, new and novel ways that are exciting and really change what we can do. So some of those fundamental changes is that data generation today is not the bottleneck. Going back just a few years, so 20 years now to the first human genome project, there were many technical hurdles, but one of the bigger ones was how to take all the sequence data and just get it in a place where it could be analyzed. Today, that's trivial. We can do that at scale. Most data now really start digital first. And something that goes along with this is that we realize that the older statistical models where we could really assume multiple variables were independent of each other isn't adequate because biology itself is highly interdependent. So we have to have much more sophisticated statistical models for analyzing things. And another point that I try to make whenever I talk about these things is that project management is critical for scaling any kind of science, and certainly the science we're doing. But like has always been true, well-defined experiments are still a key, and that doesn't go away. So I want to just start on a little vignette here and talking about patient trajectories. I think it's been very easy to lose sight of the fact that when we look at things like TCGA, which is a wonderful resource, and hopefully everyone knows what TCGA is, the Cancer Genome Atlas. It is really a snapshot of a single tumor at a single point in time with a little bit of clinical information around it. So it's, if you will, trying to understand the complexity of biology, it's a start, but it's really a very small start. So to get towards that patient trajectory, ideally we start having at least a whole series of measurements. Look at where, if you will, that line of streetlights, of lampposts, is really each measurement we're taking. And we see more and more projects like HTAN that are now taking these longitudinal snapshots of biology and trying to piece that back together. Of course, what we really want, and what I said at the beginning, is we really want to have this continuous view of ourselves, our world, and everything that actually impacts our health. So that's, if you will, the unifying vision for all the things we want to be able to do. I also want to emphasize that biology isn't just DNA. DNA is incredibly important, obviously, but understanding the role that DNA has requires actually understanding a lot more than just DNA. And this XKCD comic, I think, does that very nicely. By the way, something I didn't mention is that all my slides are available as a downloadable slides from SlideShare. So please feel free to download them, because then you can look at them on your own leisure. So I think following that point, it's more than just DNA. It's really important to recognize that there's both multiple time scales. So again, this dynamic view of biology, and size scales that make up every one of us, every organism. And that part of what we're trying to tease apart is the dynamics and the interactions between all these pieces. That's what makes both us work from a biological standpoint and makes cancer so difficult to understand, because cancer really is going in and changing some of those parameters, changing the way that some of these very interactive systems interact with each other. So something else that I think is really important, at least to put in context, is we actually don't really spend a lot of time understanding what normal is in a given cell. And so I'm going to come back to that with some of the sister projects for HTAN, because they are looking at normal. I think that's incredibly important, that we actually have a good understanding of what a normal cell is and a normal tissue is before we start trying to understand, in fact, how has cancer manipulated that normal state. So I just to look at this from a little different perspective, is precision medicine is really trying to help us understand that complex interaction between phenotype, our genes, and our environment, and how those things work together. So there's lots of different ways to cut this, and I won't go into all of them. However, again, for I would say for a data science group, it's really important to recognize that data, or what's labeled here as information, really is a collection of things, and we have to put it in some kind of order in order to have knowledge. And this little cute little graph represents the knowledge we're trying to extract from all that data. And again, I find that incredibly important to come back to again and again, is what is it how do we extract knowledge from all the data we're generating? So I want to flip back now just to talk a little bit about technology. So cryo-EM, so cryogenic electron microscopy, has been it's both an old and a new technique, and it's really enabled us to get a completely different view of things that previously really we could only see this level of detail with crystallized proteins. They're really in a very rigid structure. So what's exciting about cryo-EM is you can start to see proteins and actually other molecules in things like lipids. So again, to bring in a little biology into this, most of the important interactions, maybe I shouldn't say that, a lot of important interactions happen at membranes. They happen in really in the in-between places in the cell. So whether we're talking about the Golgi, the ER, the cell wall itself, there are lots of signaling mechanisms that work across those membranes. And we've always been somewhat blind to them. So cryo-EM's really allowed a whole new way of starting to take apart important parts of biology from a chemical structure standpoint and from a dynamicism standpoint. Likewise, single cell techniques, and I am sure that Nadia can tell you in a lot more detail than I can how some of the analyses work for single cell techniques. But single cell techniques let us look at individual cells and populations of cells in a given tissue and do it in some really very powerful ways. And so this is just a collection, if you will, of different technologies that have evolved in the past 10 years or so that allow us to look at populations of cells down to single nuclei to single cells. And along with that, and this is a really important point, is there's this ability to focus on dynamics. So how is it that these things change with time, change in response to environmental pieces? So now I'm going to bring in two different basic science problems. The first one's going to focus on something I was involved with when I was at NCI, the RAS project. And the second one will involve HChan itself. So I promised I'd get to HChan. So the first one I want to talk about is the Nci Ras Initiative. So this is really a recognition that RAS is mutated or involved in roughly 40% of all the incurable cancers. And we really didn't have a way to target it. So this is the RAS Initiative was really trying to understand some very basic biology, some very basic chemistry and biochemistry of RAS, with the hope that then we'd be able to do a better job of targeting it. So the part that I was involved with was bringing in the physics community, represented by the Department of Energy, into modeling the data that was being generated through the RAS Initiative, and then rapidly iterate between the simulations and the modeling that the computational folks were doing with the people who were doing the laboratory science. So just some facts about RAS is, again, it is a membrane-bound protein. It's an oncogenic switch. Actually, normally it's a switch. So it's either bound to GTPase and it hydrolyzes GTPase to GDP, and that's how it switches on and off. So there were a whole series of really detailed, if you will, mini projects trying to understand different parts of the biochemistry and the interactions of RAS with the membrane, and then RAS with RAF and RAS with some of the other proteins that it was interacting with. And to do that, they had to do some really interesting computation. So understand how to take things that were very detailed molecular dynamics simulations, do something called coarse grain molecular dynamics, and then bring it into a model, a phase field model, which can model much larger areas. So if you will, whole portions of a cell. And again, start to make predictions from that modeling piece, and have the experimentalists be able to either validate that they're getting part of the model right, or try new experiments and try new models. And so, sorry, there's too many little animations there. So I'm not going to go through all the details, but there were a lot of computations that were done that were really, frankly, pretty amazing, and they learned an awful lot in the course of a few years. And to make that happen, they used some of the biggest computing resources in the world that DOE manages. So again, that was if you will, a tour de force application of computing, of physicists, of computer scientists, the wet scientists doing biochemistry, doing crystallography, doing cryo-EM, and generating all that data and trying to build these more holistic models. And to make all that happen, you really have to have team science approaches. And again, that project management piece I mentioned before. I'm not going to go into this, but just so you can see the equations that they used to solve just the one piece, which was the lipid bilayer itself. And may think of lipid bilayers as being simple, but it turns out that they're actually very complicated, and the composition of those membranes really matters a great deal to things like RAS. So I'm not going to go into this one at all. Now I'm going to switch and talk about H10. So I don't know if there's questions up until this point.
Yeah, I guess I have a question. So when you're dealing with managing these sorts of huge projects, I know this is a broad topic, but do you have specific project management strategies you use?
Oh boy, well, again, I'm not the project manager, and I've worked with many different project managers over my career. And what I've appreciated is the folks that have real project management training, they have a lot of tools at their disposal to make sure that people, one, articulate what it is that they're trying to do. Sounds kind of obvious, but actually in these bigger projects, it's complicated to do that because you have to mesh different people's timelines together in order for the project to be successful. And basically hold everybody accountable and actually get them better at estimating what they're capable of. So it's really fascinating to see how a team, when it's first assembled, is, if you will, pretty chaotic. And as it starts to work on a given project, people come together and they understand each other and their strengths and weaknesses. And I would say that is, believe it or not, really what the project manager does. They make sure that people come together and work smoothly toward a goal. So again, there's lots of techniques for making that happen, but I really think in science it's incredibly important for us to do that when we have projects that require particularly multiple disciplines and large groups of people, often, and today this doesn't seem unusual, but remote. So I think that was early on a difficult thing to manage for all these remote teams.
Thank you. We don't have any other questions yet.
Okay, I've got one of my kids is running the water here for a minute, so I think I can talk again. So let me keep going, and we'll go through H10 now. So just to give you a little bit of background on what H10 is, it's looking at a number of different sites. Sorry, The 10 sites I mentioned here are the physical sites. So there are 10 different locations that are part of HTAN. And they're looking at things like precancerous lesions. So before cancer, there are often injuries, molecular injuries that happen that make it more likely for cancer to start. Doing a lot of molecular characterization, obviously using techniques like single cell transcriptomics, subcellular imaging to figure out where all this is and mapping it, proteomics, microbiome, understanding the cancerous lesions and doing some of the exact same techniques with cancerous lesions as well. And then the goal for HTAN is really creating this integrative predictive modeling environment that crosses both time and spatial resolutions. And just to go back to this slide, it's really, if you will, trying to build this kind of integrative map, but now doing it for cancer. So just in April, so 2 months ago now, HTAN had its first publication, and it's really talking about what is HTAN and what are the different projects inside of it. And I encourage you to go read the paper if you're interested. And you can see right off the bat that there is a lot, and actually the list goes up the bottom, of different organizations that are part of HTAN. I mentioned there are 10 sites, but there's far more groups engaged in HTAN than just the 10 sites. It's an impressive array of individuals across the cancer research spectrum. So what is it that HTAN is working on? It's really, I already gave away some of the punchline, and that is trying to understand those precancerous lesions, how they lead to malignancy, what triggers metastasis, and at the same time, looking at this now from a treatment standpoint, what makes a tumor responsive to treatment and what makes it resistant. And as this graphic shows, there's really, there are two different models for resistance. One is that there's intrinsic resistance. So when you give treatment, a few cells that are already present in the tumor are selected for. So if you will, that's clonal selection. And then in other tumors, there isn't a resistant cell, but you give it treatment and it evolves. So that's then clonal evolution. And again, the other way to think about that is one is intrinsically resistant and the other is an acquired resistance. So looking at HTAN from a cell type standpoint, you can see that it does focus on single cell. There are other atlases out there. There's both a human disease atlas, not cancer focused, that are doing single cell, and just as and equally importantly, there are a number of atlases that are focused on the healthy tissues. And I already mentioned why that was important, but I'll come back to it in a minute. And then of course, looking at bulk sequencing like TCGA and ICGC, they're probably the best known cancer resources for bulk sequencing. Then doing some kind of spatial analysis, so knowing exactly where you're pulling cells from. Again, to do single cell appropriately, you need to have that spatial piece. Understanding and being able to compare with healthy tissues, there's a little bit of that in each of these projects. Obviously, that's what the healthy human cell atlases are all about. Of course, looking at disease tissue and looking at detailed clinical data. And so again, that's where TCGA, unfortunately, at the time it was done, it did not capture nearly enough clinical information. So part of the Human Tumor Atlas, and I'll hopefully get to that part, is really how do we build up this detailed clinical phenotype for all the patients and understand actually how their clinical phenotype changes just as the biology of their disease changes. So here's some of the ways that we're going about characterizing individual tumors in HTAN. You can see there's molecular characterization, there's structural, and this is ultrastructural, so if you will, Some of it is subcellular and suborganelle even structural features. Of course, getting the cellular features correct, histology, anatomical features, and then the clinical features again. And being able to measure as we do all that, what's the quality of the data, and where are the data quality limitations of data that's being captured. And then really importantly, which cells are present and what are their changing and evolving roles as cancer progresses. And trying to bring in that spatial piece and that interactive piece. And that is the idea of this integrated atlas. And the hope is that not only can you integrate all that at a particular point in time, but you can also see how it changes with time and over different time spans. So that's a very ambitious goal for the Human Tumor Atlas. And again, just to show those data types, I won't go through it again, but these are the detailed data types that are being used to generate some of the spatial and molecular data. And then here are some of the organs and the diseases that HChan is explicitly looking at. So you can see from for instance, looking at breast cancer as an example, there are precancer lesions that are being looked at, primary, metastatic, and resistant lesions being looked at by different sites inside of HChan. And that's primarily, actually that's exclusively an adult. And you can see that there are a few pediatric projects as well in HChan. And then it's a matter of how do we go about putting this into this clinical trajectory. And I won't go through this slide in much detail, but just to say that this is a different way of cutting all the things that we've been talking about and organizing them in slightly different ways. And of course, each way that we organize it, we want to be able to query it and assemble the data from each of those views, if you will, of biology that's present in HChan. So now looking at it from a organizational standpoint, to make all this happen, HChan has been broken into 4 big working groups. The first one talking about policy. So how do we make sure, one of the pieces of HChan is making sure all of the data, all the tools, really all the different working products of HChan are as widely disseminated and available for everyone to be able to use. So that goes into the policy working group. And it was things like making sure that we could get open access publications from all the journals we wanted to publish in, and that was really covered for the HChan network. The clinical biospecimen working group, that's a working group I'm part of. That's really making sure, again, that we're properly defining and asking for the clinical data, the biospecimen data, the processing data on every sample that goes through HChan, and doing that in a consistent and harmonized way. Then a molecular characterization working group. So how do we actually take, for instance, the RNA that's being generated from a particular specimen and make sure it goes through the right kinds of single cell RNA-seq, if that's what we're trying to do, or the right different types of characterization, including imaging. It says molecular, but the imaging is in there as well. And then the data analysis working group is just really trying to put all those pieces together. So that's again from an organizational standpoint, how HChan itself is organized. I don't want to go too far into the details of the clinical biospecimen working group, even though I could go into that for more than an hour all by itself. But what's been interesting, and again, this is the focus on trying to do this in an open and collaborative way, is we've used the RFC mechanism that the internet itself uses for changes to the internet spec. So there are RFCs for all the different data activities inside of the data harmonization and clinical working groups, and making sure that, in fact, everyone in the community gets a chance to comment on those RFCs before they're finalized and then used to capture data by each of the sites. So it sounds a little bit complicated, and again, it is, but that's where those project managers are so incredibly important to make sure it actually works. And all of the protocols themselves, so this one is frozen tissue dissociation for single nucleus RNA-seq. That happens to be in protocols.io. Actually, all the protocols are in protocols.io for HTN. And you can go and look and actually see exactly how any given specimen was prepared, how it was handled, how it was analyzed, how the data from those analytes were generated. So here's another one from Aviv Regev's lab, looking at single cell RNA-seq expression data. Again, this is in protocols.io, and you can see it's very detailed. For instance, here, how to actually use genomespace.org. There's a list of how to actually build a recipe and which particular programs you need to install, and then it goes on. It's very rigorous in specifying all the steps you need to go through to do the same RNA-seq, single cell RNA-seq analysis that the Regev lab did. So again, that's looking at some of the things that HTN has been doing. I'm here at Duke involved in Shelley Huang's DCIS and breast precursor lesions project, along with Rob West at Stanford, and actually a lot of people at a number of different institutions. I want to give you a little bit of science background of where we are right now, and then some teasers about things that we think are going to come out of HTN in the near future, around DCIS in particular. So, sorry, just to define DCIS, it is a precursor, or it's considered a precursor to breast cancer. What's interesting about DCIS, though, is it doesn't always progress. So some women have DCIS really for their whole lives. It never seems to progress, and other women, it progresses rapidly. And we don't understand the difference. So that's one of the big puzzles that we're trying to unpack. And this just shows a group not actually part of HTN, or rather the work was not part of HTN, done by a number of folks at MD Anderson, doing laser capture microscopy, and then looking at single cell data and trying to understand which cells would go through this DCIS to invasive malignant transition. So they were teasing individual cells apart and looking at the characteristics of those cells. So very interesting project. And you can see that in this set of slides that there really was this both collections and clones that were already present, and then this selection for individual cells that could become invasive. And they did a very elegant job of showing some of that, and being able to show they could employ single cell sequencing in this very topologically focused way. So stepping back a little bit, one of the big questions is, what's the role of tumor infiltrating leukocytes, or TILs, in breast cancer? And it's been shown that inflammation is really important. But most of the work up until now has all been done with bulk sequencing. This is actually a bulk sequencing paper, but they characterized the different cell types in that bulk sequence. So here you can start to see that in fact the way they did this is that they saw that Tregs and high Treg was associated with recurrence regardless of histology. So that's important because that says at least it's likely that something that high Treg expression is doing is important for recurrence. What that is, it's not clear from this kind of an experiment. And what was also really interesting, so this is now a waterfall plot, and if you're familiar with TCGA, that upper graph looks a lot like the mutation graphs that were done fairly early in TCGA, showing that different types of cancers had the different levels of mutations. That's actually not what the upper panel is. The upper panel is looking at tumor inflammation signatures, and you can see that they in fact do not correlate with mutation burden. So there's been a lot of discussion around the impact and why some patients respond well to immunotherapies and some do not. And one of the assumptions has been that you need to have warm tumors, so those that are immunologically responsive or warm, turning out new types of epitopes for them to respond to immunotherapy. And you can see from this correlation, it's not as simple as saying just turn up mutation rates and you'll turn up the inflammation signatures and immune inflammation. And that it's not a simple picture. Again, that seems to be the case every time we learn something in cancer. We're reminded that biology is not a simple thing. So again, that was the teaser really, and that's when we do this with single cells, do we learn something new? And do we start to see that in fact there are specific cells that are really responsible for these behaviors? And I think that's one of the big questions. And I'm hoping that in the next year or two we'll be generating some of the data that helps us answer that. So I'm now going to turn to another aspect of HCHAN, and that's how it does the data coordinating. Because this is incredibly important, and like the rest of HCHAN, it's actually it's a collaboration of numerous institutions. So Ethan Cerami is at Dana-Farber, Justin Gannay is at Sage Bionetworks, Nikki Schultz is at Memorial Sloan Kettering, and Bastien Thorsen is at the Institute for Systems Biology, and they all work together on different parts of what the Data Coordinating Center needs to do its job. You know, it says the RFCs and the metadata tagging that I described earlier, that's Bastien does that. Data ingress, so how do all of us that are generating data in HCHAN get the data into a single place so that it can be analyzed? Justin worries about that. Being able to bring in all the imaging data, and imaging, for those of you who work with different types of images, you know that there are many different types of imaging in the clinic. It's very different than, for instance, in research. Data that's coming out of ultrasound, coming out of X-ray, coming out of cryo-EM, it's all, you have to handle them all differently. So imaging is a big deal. And then how it all becomes, how we package it up and release it. And, that's Ethan's job with the portal. So again, just to focus in a little bit on the metadata, because that's more my side of things. It's really describing the clinical attributes, the clinical specimens, how all the different data files are related to each other, what's in them, and how the different experiments relate to each other. And that all really needs to be very explicit and well laid out in the data portal so other people can use the data. Something I should point out is that all the data for HCHAN goes into Synapse. That's a cloud-based data platform that Sage Bionetworks has developed. If you're familiar with the various stream challenges, those are all run in Synapse. So we believe that's a good place to put all of our data so that in fact everyone can have access to it. And again, a big part of HTAN is working with some of the other groups I've already mentioned. Maybe not by name, but Human Cell Atlas I did by name. HubMap is a biomolecular atlas project. Again, there's lots of great data that's available in those resources that we really want to be able to interoperate with HTAN to understand, again, the normal biology and then cancer biology. So the RFC process that I tried to describe verbally, here's a nice graphic of how it works. And that is, again, a central part of making the data harmonization really work for HTAN. I won't go through this, but just to show that, in fact, there's been a lot of progress since HTAN started roughly 2 years ago. We have now a lot of the RFCs have been completed. And in fact, we're getting ready to really load the first set of data into Synapse for HTAN in August. And that's really a collection of all these different things that'll be there. You can get the RFCs. This is all inside of Synapse in the HTAN project inside of Synapse. And again, there's been a lot of focus on how do we make the data deposition as easy as possible. So the idea is it goes into AWS. If you already store your data locally in AWS, it moves in pretty easily. And then there's you create a file that lists all the metadata that you're going to upload. That actually creates a validator for all the files that you actually upload. And so then the upload actually goes through and validates that it's getting what you said you were going to submit. And that's just what this slide highlights. So again, this is if you will, a very operationally focused view of the data that goes into HTAN, but it's critical to ensure that when people go in and want to analyze the data, they're actually analyzing what they think they're analyzing. Just to show the imaging plan that Nikki's been working on, the next slide is have a collection of these 3 very different types of viewers. Again, because there are such different types of imaging modalities that we want to make sure are accessible inside of HTAN. And they're for those of you who are familiar with either pathology imaging or other types of imaging modalities from CyTOF and other things. These are really some beautiful tools for interacting with all that imaging data. And going back to the spatial piece, you'd like to know when you're for instance, doing RNA-seq on a particular sample, where exactly from that slide did those data come from, that sequence data or the MIBI data or whatever it is. So again, there's a complex mapping between these different data types to make sure we can do that spatial and temporal mapping as well. So just to look at HTAN from a atlas standpoint, so the idea that there are multiple atlases. So one of the atlases that I was describing was really looking at DCIS, but every cancer type and every part along that progression can be thought of as an atlas. And we want to make sure it's easy to upload those atlases, analyze those atlases, and then also go between these different views to understand how biology is either similar or different, for instance, between different stages of the same cancer. And this just shows from an architecture standpoint, I mentioned that there's AWS behind Synapse, and then there's a portal that sits on top of it with a content management system. And this is the portal. And actually, I think on a previous slide, there should have been the URL for this portal. And again, this is still beta. The data is still getting ready to go in. So again, I'm talking about it a little bit before it's ready, but I think that's also exciting. So you all are prepared to go in and take the most advantage of what will be there. So with that, I think I am just about on time, and I'd be happy to take your questions. And it looks like at least one question came in.
Thank you very much for a wonderful talk. And yeah, we so far have one question. So how do you reconcile the implications of patients that have been treated with multiple treatments for HTAN and clinical trial outcomes, as well as interpretation?
So that's a wonderful question. Thank you. Right now, so for HTAN, we're being very, I think, very cautious in how we think about multiple treatments, particularly for earlier in the disease progression. So at least with DCIS, for instance, we're really looking for pre-treatment measurements and pre-treatment specimens, and then being able to follow exactly how that particular patient responds to the therapy they're being given. So this is not a clinical trial data that goes into HTAN, in that the patients that are getting therapy in the clinic, they're getting standard of care therapy. They're not getting a clinical trial. So your question is a good one, though. We will be characterizing, and I'm not quite as familiar with some of the other groups, but we will be looking at advanced disease, and different patients will come who will present with different prior therapies. So I think that the best answer I have for you is that all that will be tracked. So for instance, I believe we'll have a few hundred lung cancer patients that are going to be very carefully measured, and we'll know which treatments they had gone through before biospecimens were taken. So I think that I'm hoping that answers your question. And again, the way that we're thinking about both acquired resistance and the existing resistances, that would primarily be through generating PDXs and things like that would allow us to take those cells and then test them with some of those different therapeutic agents and see how those cells respond. That's different than, obviously, than doing that directly in the patient. But hopefully what we would find out in doing that would inform a clinical trial later.
Thank you. And then in pediatric ALL, a number of kids will undergo a stem cell transplant. Are you also doing these measurements on the donors, for example, related or unrelated donors?
So unfortunately, I don't know enough about the ALL project to answer that question. I'd be happy to put you in contact with the folks that are actually doing that. I don't believe they're going and getting the donor material, but or testing the donor material, or actually testing the donor. But I don't know that for sure.
And then how do you account for differences in available imaging and pathology data from different patients as you create the database?
Oh, that's a great question. These are all great questions, making me work here. So it turns out that there's both the clinical imaging that we will get and we are getting on those patients. But then we are doing very standardized, particularly from a pathology standpoint, very standardized pathology handling and imaging of every case that comes through. That's actually part of what the clinical annotations and biospecimens working group is standardizing, is exactly how to consistently measure across different tumor types, all the histologies, all the tissues, so we get consistent data. But as you're pointing out, it's really a non-trivial problem, in part because you have to handle the different tissues differently. So how you go about compensating for all these different tissues is itself really complicated. So again, part of the answer, of course, is just being able to annotate exactly how it was done and from which tissue. But it's also about standardizing the handling of the tissues as much as possible, and of course, the way the imaging is being done. So again, that's part of what the clinical annotations and biospecimen working group is doing.
Thank you so much. So, so does your standardization affect how you recruit patients?
So we've tried to be really thoughtful in not creating what I would call the eligibility gauntlet that clinical trials go through. With that said, though, we want to identify certain types of tumor features that we want to make sure that if you will, a given atlas has. So yes, there's a selection, but we're trying to make that as specific to the cancer phenotype or the cancer presentation as possible, and not really worry about even the comorbidities present, because that again, we're not trying to run a clinical trial. So I think we have some very different capability for bringing patients into, or bringing their tumors into HTAN. So I didn't show this slide, but we actually have a really interesting look across the diversity and the SES status of patients that are donating samples to HTAN. I've been very pleased to see how representative that is of each of the catchment areas for each of the sites that are doing that are getting those donors. So I believe we're going to do a good job of representing the diversity of patients that we see. But of course, we want to pay attention to that.
Any other questions? Well, thank you again.
Yeah, thank you for the great talk, Dr. Kibbe. And I would like to thank you all for joining us today. So this is the first year of our Big Data Summer Workshop. Hopefully next year we will be able to have the hands-on session on campus. So have a great weekend, and hope to see you next summer.